Hello, my name is Rayana Anthony, and my group was experimenting the effects of pH on trypsin activity. So, a little introduction. So the enzymes are catalytically active biological macromolecules. So that means that enzymes will help speed up the chemical reaction by lowering the activation energy. Um, so we'll do this by bonding to the substrate and then creating microenvironments that promote the reaction. And then our abstract is that trypsin is a protease that breaks down other proteins. It's found in our digestive system. As stated, the role of trypsin is to break down proteins. Once the proteins are broken down, they are now small peptide chains and amino acids. So we decided to test the efficiency of trypsin on azocasein at a pH of 7, 8, 9, 10, and 11. And originally we did test it at a lower pH because we assumed that trypsin would work better in a more acidic environment, but ultimately we found that trypsin works better in a more basic environment. So we figured that trypsin would be most efficient at a pH of 8, and we ran the temperature at 37 degrees Celsius, which is body temperature. So we believe that this will be the most natural environment for trypsin to react in. And based on our results, we concluded that although there were no real significance between the data, our hypothesis was correct because trypsin still had the most efficiency at a pH of 8. And then, just to reiterate, our hypothesis was that a pH of 8 will provide the most efficient environment for trypsin to break down the protein azocasein. All right, and then moving on to our materials and methods. So the steps in this, we added the following to a clean centrifuge tube and ran it 3 times. At each pH, we added 450 microliters of each pH. Then we added 500 microliters of the azocasein and then 50 microliters of the trypsin. We also did run 2 negative controls, which consisted of water. So instead of using the trypsin, we used water to see what the reaction speed would be at if there wasn't an enzyme. Then we incubated for 10 minutes at 37 degrees Celsius. Again, that is body temperature, so we believe that would be the most efficient temperature to run the experiment at, since trypsin is found in the body. And then we use 500 microliters of 10% TCA. And we did add the TCA to the centrifuge tube to ensure that the reaction between the azocasein and the trypsin was completely stopped. So this helped make sure that the reaction and the end time, the reactions ended at the same time before centrifugation, because that would affect our results if there was a reaction still going on during the centrifuge. And then for step 4, we placed the test tubes in a centrifuge for a maximum of 2 at maximum speed for 2 minutes. And that was just to separate the broken down protein from the unbroken down protein. Moving on, we transferred the supernatant into a clean cuvette. And the supernatant, that was the broken down protein. So that is what we were measuring. We wanted to see the concentration of that to see how much was broken down. And then we blanked the spectrophotometer at 450 nanometers using a cuvette of deionized water. And then we read the absorbance of the spectrophotometer at 440 nanometers. And then just to answer some questions that might be missing, again, we added the TCA to ensure that the reaction was completely stopped. We did set negative controls, and we ran 2 negative controls at each pH to just see what the reaction speed would be like without an enzyme. We also did have replicates to lower the standard deviation. And by lowering the standard deviation, that would mean that our results were more accurate. And then we centrifuge before or after adding the TCA to ensure that the reaction was completely finished, and to to ensure that the reaction was completely finished, so that the centrifuge could completely separate the unbroken down protein, which is the pellet, from the broken down protein, which is the supernatant. And then lastly, the controls that we used was the temperature. That was the main thing. We chose body temperature. Originally, we did have 25 degrees Celsius, which was room temperature, but we ended up finding that by changing it to body temperature, raising the temperature, it did help our reaction a lot. Okay, and then moving on to results. These are our results. Alrighty. And you can see that for our standard deviations, they are relatively small. The only one that is a little large is at a pH of 7, and that was probably human error, something with pipetting. But ultimately, our standard deviations are small. And then moving on to graphing, you can see at a pH of 8, the absorbance level was high. You can see that bell curve right there. And then for our test, we found that for a pH of 7 and pH of 8, they were not significant. And then also at a pH of 8 and 9, they also were not significant. So therefore, we conclude that there was no significant difference on the effects of trypsin between pH levels. Alrighty, and then for our conclusion, we said the experiment design and execution was adequate to test our hypothesis. We had negative controls to see the reaction speed without enzymes, a pH range that clearly displays optimal enzyme activity, and several trials for each pH level. We were missing all these components in our first experiment, but due to these changes, we were able to run and execute an adequate experiment. Based on the new data, we can conclude that our hypothesis was correct. The absorbance of trypsin's highest at a pH level of 8, and at a temperature of 37 degrees Celsius, which is body temperature. The optimal pH for the enzyme activity is slightly basic. And then here are our references. Alrighty.